Most of a eukaryotic cell's ATP synthesis occurs in mitochondria. A mitochondrion is a sac within a sac. The inner sac is folded, increasing surface area for ATP synthesis. Between the inner and outer membranes is the intermembrane space, a reservoir for hydrogen ions used for synthesizing ATP from ADP. The inner chamber, known as the matrix, is a soup of enzymes that dismantle fuel molecules in the Krebs cycle. The knobs are where ATP is synthesized. Simply put, a mitochondrion is an energy transformer, where energy in fuel molecules such as pyruvic acid is transferred to ATP. Carbon dioxide, the carbon end product of cellular respiration, diffuses out, leaving the cell through the plasma membrane.
In this video, we are going to look at glycolysis. Glycolysis is the process of breaking down a glucose molecule into pyruvate and gaining a couple of ATP and NADH in the process. Glycolysis begins with the molecule glucose. The enzyme that begins this process is called hexokinase, or glucokinase in the liver. A kinase is an enzyme which adds phosphate groups onto things by taking them off an ATP. And that's exactly what happens here. An ATP donates a phosphate to the glucose and creates this molecule, glucose 6-phosphate. It seems odd to use ATP in this process because we are trying to create ATP, but we'll see that we get all this ATP back plus more once the process is complete. The glucose 6-phosphate is acted upon by phosphohexose isomerase. Isomerases are enzymes which rearrange the structure of the substrate without changing the molecular formula. We can see that the phosphohexose isomerase rearranges the glucose 6-phosphate into a fructose 6-phosphate molecule. Now we meet another kinase, phosphofructokinase 1. This enzyme attaches another phosphate group to our molecule, creating fructose 1,6-bisphosphate. Sorry, my mistake, that should read fructose 1,6-bisphosphate, not bisphosphonate. Anyway, moving on, the molecule is then split in half by fructose bisphosphate aldolase. An aldolase is an enzyme that creates or breaks carbon-to-carbon bonds, which makes it the enzyme of choice to split the fructose 1,6-bisphosphate molecule into these two molecules, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. The next step is conducted by another isomerase, the triose phosphate isomerase, and this converts the dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. The next enzyme we meet is glyceraldehyde 3-phosphate dehydrogenase. The dehydrogenases move a hydride ion, that is H-, onto an electron acceptor such as NAD+ or FAD. This is what happens here, creating two NADH. There are two molecules of NADH created because one glucose molecule created two glyceraldehyde 3-phosphate, one from the aldolase reaction and one from the triose phosphate isomerase reaction. The hydride group on the molecule is replaced by a phosphate group supplied from inorganic phosphate, not from ATP. It is the reduction of NAD+ into NADH which provides the energy to bind inorganic phosphate. This gives us the molecule 1,3-bisphosphoglycerate. Here the molecule meets another kinase, phosphoglycerate kinase. We saw above that kinases can giveth phosphate, but they can also taketh phosphate away. And that's exactly what happens here. 2 ATP are produced by taking phosphate from the 1,3-bisphosphoglycerate molecules. This produces the molecule 3-phosphoglycerate. The enzyme phosphoglycerate mutase is essentially an isomerase, so it creates the isomer 2-phosphoglycerate from 3-phosphoglycerate. Enolase is the next enzyme, and it produces a double bond by removing the hydroxyl group. This leaves us with phosphoenol pyruvate. The final enzyme of glycolysis is pyruvate kinase. And this kinase is also going to remove phosphate groups from the molecule, attach them to an ADP, and create ATP. Again, 2 of these are created because 2 phosphoenol pyruvates will be created for every glucose molecule. This finally leaves us with pyruvate, which will be the substrate for the TCA cycle. Now for your reference, I shall quickly number these steps, and you can see that there are 10 steps in total. Steps 1 to 5 are often referred to as the investment phase because they use up ATP. Steps 6 to 10 are known as the payoff phase because we get back our initial
Glycolysis is one of the pathways cells use to transform sugars like glucose into biochemical energy in the form of ATP. In the cytosol of the cell, glycolysis converts glucose into pyruvate through a series of 10 enzymatic reactions. This process produces ATP, along with other products such as NADH, that can be used later to produce even more ATP for the cell. Let's watch as these enzymes oxidize one glucose molecule into 2 pyruvate molecules. First, a kinase reaction adds a phosphate onto glucose to form glucose 6-phosphate. This is one of 2 energy consumption steps and is an irreversible reaction. Next, an isomerase reaction converts glucose 6-phosphate into fructose 6-phosphate by rearranging covalent bonds. Another kinase removes a phosphate group from ATP and gives it to fructose 6-phosphate to form fructose 1,6-bisphosphate. This is the second energy consumption step and an irreversible reaction. In the fourth step of glycolysis, a lyase reaction splits the 6-carbon fructose 1,6-bisphosphate into 2 3-carbon sugars, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. The dihydroxyacetone phosphate is rearranged by another isomerase to form a second glyceraldehyde 3-phosphate. At this point in glycolysis, glucose has been metabolized into 2 glyceraldehyde 3-phosphates and 2 ATPs have been consumed. The next 5 steps of glycolysis are the energy-producing phase. In step 6, both glyceraldehyde 3-phosphates are oxidized to 1,3-bisphosphoglycerate by a dehydrogenase. This step produces 1 NADH for each oxidized glyceraldehyde 3-phosphate, for a total of 2 NADHs. These NADHs are later used to produce more ATP for the cell. In step 7, a kinase transfers a phosphate from 1,3-bisphosphoglycerate to ADP to form ATP and 3-phosphoglycerate. This step is reversible even though ATP is formed. The next step involves a mutase reaction that moves the phosphate on the 3rd carbon of 3-phosphoglycerate to the 2nd carbon position to form 2-phosphoglycerate. In step 9, a lyase reaction removes water from 2-phosphoglycerate to form phosphoenol pyruvate. In the final step of glycolysis, a kinase reaction removes the phosphate group from phosphoenol pyruvate and donates it to ADP to form ATP and pyruvate. Like reactions 1 and 3, this step is irreversible. At this point, 2 pyruvate molecules, 4 ATPs, and 2 NADHs are formed for each glucose that was broken down in glycolysis. The pyruvates and NADHs could be used in aerobic respiration to produce more energy for the cell. Here, we depict glycolysis as a closed process. But in cells, substrates produced by other reactions can enter glycolysis at different points. For example, when an animal breaks down glycogen, glucose 6-phosphate is produced and can then enter the glycolysis pathway at the second step. Importantly, this means 1 less ATP is required for the pathway because the first ATP consuming step is skipped. Other sugars can also enter the glycolysis pathway at different points, each having a different effect on the net number of ATPs that are produced.
The primary substrate for the TCA cycle is pyruvate. Fatty acids and amino acids can also be used, but we're not going to discuss them in this tutorial. Pyruvate is produced by glycolysis, and the first thing that happens to pyruvate is that it gets converted into acetyl coenzyme A, more commonly known as acetyl-CoA. This conversion happens via the action of 3 enzymes, collectively known as the pyruvate dehydrogenase complex. When you see the word complex after an enzyme, it usually means there are multiple enzymes involved, but they are functionally considered as one. As we saw in the glycolysis tutorial, we'll look at the molecules involved first, and then we'll discuss the enzymes involved. The TCA cycle begins when a 4-carbon molecule known as oxaloacetate combines with acetyl-CoA to produce a 6-carbon molecule known as citrate. Citrate becomes a 6-carbon molecule, cis-aconitate, and then cis-aconitate becomes the 6-carbon molecule isocitrate. In the next step, we lose one of these carbons as carbon dioxide, and we also produce another NADH. Thus we are left with a 5-carbon molecule known as alpha-ketoglutarate. The same thing happens again. We lose a carbon as carbon dioxide and gain an NADH. Hence we are left with only 4 carbons in our main molecule, which is now called succinyl-CoA. Succinyl-CoA is then converted to succinate, and a GTP is produced in the process. GTP is essentially an equivalent of ATP. Succinate is then converted to fumarate, and an FADH2 is produced in the process. Fumarate becomes malate, and malate completes the cycle and becomes oxaloacetate, producing the final NADH in the process. The oxaloacetate is then available to combine with any available acetyl-CoA and begin the whole process again. Now let's take a look at the enzymes involved. Citrate synthase predictably synthesizes citrate from oxaloacetate and acetyl-CoA. Then, the enzyme aconitase produces cis-aconitate, and then the conversion back to isocitrate, which is a structural isomer of citrate. Again, we see dehydrogenases in action, with isocitrate dehydrogenase producing the NADH in the isocitrate to alpha-ketoglutarate conversion. And the alpha-ketoglutarate dehydrogenase complex also producing NADH in the conversion to succinyl-CoA. Succinyl-CoA synthetase catalyzes the conversion of succinyl-CoA to succinate. Then yet another dehydrogenase, known as succinate dehydrogenase, produces the high-energy molecule FADH2. Fumarase converts fumarate to malate. And then the final enzyme is another dehydrogenase, malate dehydrogenase, producing NADH and converting malate to oxaloacetate. And that's an overview of the TCA cycle. In this video, we're going to look at oxidative phosphorylation, which is also known as the electron transport chain. You may remember that a mitochondrion looks a little like this. It has an outer membrane and an inner membrane. And we're going to take a close-up look at the inner mitochondrial membrane. So here is the membrane, and within it are a bunch of protein complexes. They are known as complex 1, or NADH dehydrogenase, complex 2, or succinate dehydrogenase, complex 3, or cytochrome C reductase, and complex 4, or cytochrome C oxidase. These are the major protein complexes of the electron transport chain. The other factors involved in the electron transport chain are coenzyme Q10 and the cytochrome C complex. The other large protein complex involved here is called ATP synthetase, and as the name suggests, it is the complex that makes ATP. So the process begins with NADH, which was produced by glycolysis, beta oxidation, and the TCA cycle. This NADH is oxidized to NAD+ and donates 2 electrons to the electron transport chain. These 2 electrons get passed between the complexes like this, and are finally accepted by oxygen, and with a couple of protons, this produces water. This oxygen is supplied by breathing air from our atmosphere, and to provide this is the reason that all animals need to breathe. Now, as the electrons are passed between the complexes, they pump protons out of the mitochondrial matrix into the intermembrane space, like this. Complex 1 pumps out 4 protons, complex 3 pumps out 4 protons, and complex 4 pumps out 2 protons. Therefore, in total, one NADH pumps 10 protons across the membrane. A similar process happens when FADH2 is used as the electron donor. 2 electrons get donated to complex 2, which in turn passes them to coenzyme Q10 and onward down the electron transport chain. Because complex 1 is not involved when FADH2 is the electron donor, only 6 protons get pumped across the membrane. Finally, these protons then flow back down their electrochemical gradient through the ATP synthetase. For every 4 protons that flow through the ATP synthetase, a single ATP is produced. I should just say that different textbooks will quote different numbers here. I've quoted the number from Leninger's Principles of Biochemistry, 5th edition. So if you do the math, it turns out that one NADH will produce 2.5 ATP, and one FADH2 will produce 1.5